Hello everyone. In the previous video, we have studied about what is cell, right? The history of cell and the components of cell. In today's short video, we will be discussing a part of the first component of cell, that is cell boundary. Now, we have studied that this cell boundary have two parts. One is your cell wall and the second one is your plasma layer, right? Now, this term cell wall was first given by Robert Hooke. In this slide, the spelling of Hooke is mistaken. It should be Hooke. Now, we have studied that this Robert Hooke, for the first time, observed cork tissue under a microscope where he observed cell for the first time. Now, in this cork tissues, all the cells are dead. Now, what did then this Robert Hooke observe? He observed the cells which didn't had any cytoplasm or nucleus, but only the cell wall. Okay. Now this cell wall is non-living rigid core which surround the cell. Why it is needed? It is needed to give any shape, a shape to the cell. Okay. And it also provides mechanical support and strength to a tissue and an organ. So for example, let us take an example of egg. Okay, and let us consider that the egg shell is the cell wall of that cell. Okay, now if you remove the shell from the egg, how will it look like? It won't have any shape, right? It will just spread. Therefore, why cell wall is needed? It is needed to provide shape to a cell, and on the basis of structure of this cell wall, tissues are identified. Okay, there are several kind of tissues in a plant. Okay, now on the basis of this structure of cell wall, the tissues are identified. Again, in this cell wall also there are three parts. One is your middle lamella, second is your primary cell wall, and the third one is your secondary cell wall. Okay, now let us first discuss about the middle lamella. You can see in this diagram that it is the topmost layer, or it is the topmost part of the cell wall. Okay, now what does it do? It acts as an adhesive between two cells. That means it joins two cells together. To form any kind of structure or any kind of tissue, cells should be joined, right? Now this plasma lemma acts as an adhesive to join two cells. Okay. Now, primarily, this plasma lemma is composed of pectin in the form of, which is present in the form of calcium and magnesium salts. And this pectin can be dissolved by an enzyme named pectinase as well as any kind of strong acids. Okay. So, for example, if you take any fruit like mango or banana, you will notice that when the mango is immature, okay, it is very hard. Why it is hard? Because of presence of pectin. Okay, now gradually when this mango matures, it produces certain kind of acids, because of which this pectin, this layer of pectin or this middle lamella loosens because of degeneration of pectin. Okay, now most of us, those who have studied a little bit of biology, must be having this conception, right? That cell wall is made up of cellulose. But let me tell you this, that in middle lamina, cellulose is absent. Further, when we will be studying the cell division, we will also find that this middle lamella is the first to originate just after cell division. That means just after cell division, this middle lamella starts to form. Okay. Now let us talk about primary cell wall. So this primary cell wall lies between middle lamella and plasma lemma, and it is mainly composed of hemicellulose and cellulose. Okay. I think hemicellulose is around your 56%, and 53%, and cellulose is around 30%. Okay. And apart from this hemicellulose and cellulose, there are other components also like pectin, protein, and lipid. Now, what is the cellulose? The cellulose is a fiber-like structure which is formed of around 3,000 glucose molecules. That means 3000 glucose molecules, they combine together to form cellulose. Again, this cellulose again combined, that is 2000 molecules of cellulose, they combine to form the cellulose microfibrils. Okay, these microfibrils are the this cellulose microfibrils are the unit of cell wall structural organization. Okay. Now, in case of primary cell wall, this cellulose microfibrils are loosely arranged. That means there will be some pores in the primary cell wall due to improper deposition of your cellulose microfibrils. Okay. Now, these pores are known as your plasmodesmata. Okay. These are called plasmodesmata. Okay. Now, these plasmodesmata, what they do, they act as a channel between two cells. Okay. Now, one more information about this is the meristematic tissue contains only the primary cell wall. Okay. Now, let us talk about the secondary cell wall. Now, this layer is the last layer to be deposited in the innermost wall of the cell wall. It lies between the primary cell wall and plasma lemma. It's composed of cellulose, mainly of cellulose, and others are your lignin, suberin, and lutein. Okay. And in this case, the important thing is that the microfibrils are arranged nicely. That means throughout this secondary cell wall, the microfibrils are arranged properly. That means there is no bone formation in case of your secondary cell wall. So this is it for today's video. In the next video, we will be studying about the plasma lemma in detail. Thank you very much.